Brilliant. Well, thank you so much, everybody. It's been absolutely amazing festival. I've learned so much. But we have one more talk, and it looks really interesting. So is it Saeeda who's going to? Is that right? Am I getting my?
Saeeda, yeah.
Yeah, Saeeda. Yeah. Okay, yeah, brilliant. Well, I introduced Saeeda to talk about science, communication, and genetics from medical school to primary school,
Thank
which is
you.
an excellent way round, I think. That's exciting me already.
Okay, are we sharing?
You're not sharing it? Yeah,
All right,
fantastic.
okay, perfect.
Brilliant.
Okay, so just want to say hello. Thank you for joining us, especially because it's at the end of the day. So I'm Saeeda, and I'm here with Doha and Esther, and we're from the School of Medicine at the University of Glasgow. Now, we're here today to share our experiences of our project, Science, Communication and Genetics, and it's from medical school to primary school. And I'm going to begin by giving you the background of the project. Doha will talk about the student perspective, and then we'll finish off with a fun activity led by Esther. Next slide, please, Doha. Okay, so as part of the medical school curriculum, students can choose a module that aligns with their interests. This is called the student selected component. And this SSC was selected by 6 medical students, and it focused on teaching genetics using the active learning approaches which are used in science communication. And the SSC, it ran over 5 weeks, and with 3 of those weeks spent in a local primary school. So week 1 was the introductory week for the medical students, and that's where the core genetics concepts were reviewed. And they also attended an in-depth lecture on sickle cell disease and explored its cultural and societal impact, and that was using Nigeria as a case study. Now, additionally, the students received, that was the first week as well, so they received a briefing for the school project during this week as well. Then weeks 2 to 4, that involved 2 afternoons a week in the primary school, delivering lessons and also learning about other science communication approaches, things like podcasts, videography, blogs, there's different kinds of approaches that we can use. And then finally, week 5 was their assessment week. So the SSC was designed to align with the Scottish School Curriculum for Excellence and the Year 2 genetics from the medical course. So really it was finding that point where the two could be combined and building on existing knowledge for both the primary school children and for the medical students, where they could both kind of work together. And then it also connected back to the University of Glasgow Graduate Attributes Framework, which outlines core skills that students are to develop over time. Next slide, please, Dora. Thank you. So the information was presented to the children as a story. So it was a modified PBL approach that we took, so the problem-based learning approach, which is quite common in the medical school at Glasgow University. And we began by exploring the Eat Well plate. Now, the Eat Well plate is the children in the primary school, they're introduced to that probably when they're in P4, so maybe about they're 8, 9 years old. And what they were asked was that they already knew about anemia. So they knew that there was nutritional deficiency anemia, and then we led into sickle cell anemia, and the latter was what was being inherited. And to make the concept of the circulatory system more accessible, it was turned into like a playground game, which Doha is going to talk about later on. And this playing approach, it led into discussions on the components of the blood and the differences between healthy and sickled red blood cells. And it was at that point, and when we went back into the classroom, the children also built models of blood cells using Play-Doh. Now, since many of these concepts, they introduced new vocabulary, there was spaced repetition, so students were constantly reinforcing these concepts and asking them to note these down. And there was also visual aids as well. And at the point where we came to genetics, the children were already aware of this from things like the TV and just hearing it around the concept of genetics and DNA. And this was expanded on by introducing the concept of inheritance using a hands-on activity using juice bottles. Then all this information was tied together by explaining the concept of selective advantage, and that was to show how sickle cell trait can offer protection against malaria, particularly in warmer climates. And then what the students also did was they did presentations in games, hands-on activities, and group posters, which were for their parents and the P7s. So I'm going to pass you over to Doha, who's going to give you the student experience.
So hello everyone, my name is Doha. I'm a second year medical student at the University of Glasgow, and today I'll be taking you through what it's like being part of the Science Communication and Genetics SSC. So first I'm going to talk about what I had in plan, specifically why I chose this SSC in the beginning. So a bit about myself is that I am interested in child health and well-being. So when I saw the module description of this SSC, including working in a primary school, I was very excited to have an opportunity to work closely with children and further develop my communication skills with a younger age group. So during this SSC, I had the opportunity to have one-to-one and group discussions with the children, which helped me understand their communication skills and the different non-verbal and verbal cues they use to express their thoughts, interests, and opinions and topics. This could be their facial expressions, body language, or frequency of participation in discussions. And as you know, communication skills is a vital skill that doctors have to be or to be able to perform and be able to give optimal patient experiences as well. So I'm very grateful to have had developed my communication skills in the most appropriate environment and with the most appropriate guidance as well. Another reason why I chose this SSC was because I wanted to improve my public speaking skills. And as for the longest time, I've been avoidant of public speaking and presenting in front of an audience. But when I saw this SSC had an assignment of a presentation, I thought it's going to be the perfect opportunity for me to get out of my comfort zone. And it was. So these are some pictures of my presentation with my peers on sickle cell anemia and malaria. And as you know, it's really hard to keep children engaged for a really long time when you're just talking. So we thought to make the presentation more engaging, to have different activities and visuals for them. So instead of explaining concepts verbally, we got to have visuals and we got to play spot the difference games. So as you see, the slides had the structure of red blood cells, the biconcave red blood cells, and the sickle cell. And we got them to spot the difference and explain what is the difference between them. And we also got to have a quiz at the end to test their knowledge. And we got to play stand up for true and sit down for false, which kept them engaged in answering the different questions we had for them. And it made the presentation more enjoyable for them and for us as well. The last reason why I chose this SSC was because I wanted to be part of a big impactful project, as this SSC was designed and executed in Scotland, but it was aimed to be translated to primary schools in Nigeria. So during our time in the SSC, we got to work closely with medical professionals and students as well from Nigeria, and they got to tell us about their culture and different healthcare system they've got there. So it was very interesting and insightful. I'm looking forward to our project, making it across the world and making a difference. And now moving on to what I didn't have in my plan and got to learn during the SSC was to know what it's like to be in a role of responsibility and taking ownership. So during the time of SSC, I got a child with additional support need join my group very last minute. So I had to be very adaptable in making changes to the activities we had to make it more accessible to the group and suit his needs during his learning process. So some of the activities we had for him were very hands-on activities. We got to play with Play-Doh and make red blood cells into sickle-shaped and biconcave shaped. We also got to make a figure of a human body and had him like trace his fingers around the body to recreate the circulatory system. We also got to learn to play around with packaging foam and color it in different colors. So for the posters, we got him to color it blue and green for Nigeria and Scotland. And he also made a sickle-shaped packaging foam as well that he got to present during the poster presentation at the end of the SSC, which the audience really enjoyed just playing around with. So another thing I got to learn was how much the children can comprehend and retain. To be completely honest, at the beginning of the SSC, I had no expectations for the children to actually comprehend the medical and scientific topics that, we get to learn about in medical school. So like imagining a 10-year-old explaining genetic inheritance was just really hard for me to actually like imagine. So and I think the reason behind their comprehension and attention was because the children were actively engaged with the material they had. And we also incorporated spaced repetition where we got to quiz them on the material before and after each session we had with them. So now some of the examples we had of the activities is that this activity of inheritance patterns where we got to use juice bottles to recreate genetic inheritance. So this example is a sickle cell trait of an autosomal recessive genetic inheritance. So at the back, we've got both of the parents being carriers, and at the front, we got the children. So on the left, we got two carrier children that got one pair of the gene. And then we got an affected child which got both of the genes, which means the child has sickle cell. And then on the far right, we got a child that didn't inherit any of the genes, so he had normal genes. And this helped the children visualize how genes get passed down in between generations. And it also helped me in reinforcing the existing knowledge I had of the genetics as well in teaching them. So another example we had was problem-based learning, which is something that we used in Glasgow's medical school. And we had a modified version of it for the children to suit their learning abilities. So we got a scenario of a child who had a nutritional anemia. And in between different concepts, we got prompts and activities for them to aid their learning and to also use their critical skills and problem-solving skill. So this is the playground activity we had where we recreated the circulatory system. We had the children just run around the playground as red blood cells. And then we got to recreate a sickle cell crisis where we told them to just stick together and reform like an occlusion of a vessel. And this helped them visualize what is happening inside of a patient's body during a sickle cell crisis. So the last and most important thing I got to learn during the SSC is to be open-minded and learning from others. As during these 3 weeks, I got to teach the children, but I also got to learn a lot from them. And it was a really good experience with them, and I appreciated a lot. So this is an appreciation slides of all the posters they got to present for their teachers, older children, and their parents as well on the last day. So now I'll be passing you over to Esther.
Hi guys, so I'm Esther, and I'm an alumni from Glasgow University. So I've done my master's in medical genetics. I graduated last year. So when I graduated, like most people, I just didn't know what I wanted to do. I just knew I didn't like the lab. That's all I knew. And I was trying to figure out from there what else I could do. So during my master's, Saeeda was actually my supervisor, and we got to talking and things like that. And she was really involved in science communication and public engagement and things like that. And from speaking to her, I started to understand that maybe that's kind of the route that I wanted to kind of go into. So she invited me along to help her in the SSC, which was a great opportunity. And I came in as somewhat of like an evaluator and just to lend a helping hand where possible. So Glasgow Uni has a set of graduate attributes that Saeeda mentioned before, and those are just like the personal qualities and transferable skills that they want students to have the opportunity to develop. And so they can take that into employment, but something I've also found is like you can just take that into life itself. So coming into the SSC, I realized that I was demonstrating some of the skills I had learned. So skills like the confidence, the effective communicator, and adaptability. I feel like all these three are very important, especially when we're working with kids. I think these are the three most important ones I picked out of it. And it just showed how much these attributes that I learned in university actually helped me towards active learning and helping out with the kids and things like that. So we're going to move on to the interactive activities a bit more now. So I don't know if you want to turn off the recording now.
Yeah, we can pause the recording. Yep, sure.
It's just so everyone feels a bit more comfortable. And if you don't have any scissors, it's fine. You can do a little bit of a tear around, whatever works for you. So we're going to be making some sickle cells and normal red blood cells. So I'm going to get you guys to draw them out. So for the red blood cell, this kind of brings back the icebreaker that we've done. So I told everyone to get a banana. So most of the kids, when we asked them what the shape of a sickle cell was, the first thing that came up to mind for them was a banana. So in the slide, we can see the little pictures on the side, and that's just an example of the sickle cell over there. And I also want you to draw a normal blood cell as well. So again, when we were playing the game, I asked you to get a plate because a lot of the kids, again, associated the normal blood cell with like the plate or the donut, that kind of shapes, like things like that. So again, there's just a shape on the side just to give you a little example so you're able to draw that out. And once you're done doing that, I want you guys to color them in and just cut them out, and you can label them in the back. And I'm going to let you guys keep doing that. I'm kind of just going to keep talking at you, and so just when you're ready, I just want you to put them up whenever you're ready. So these are all very good examples that we found of active learning when we got the kids to sit down and actually draw out the shapes and draw it and describe them to us. Again, as Doa mentioned, we used a lot of Play-Doh as well, and getting the kids to shape out the cells was a good way for them to actually learn it. And then it's a repetition of looking at the different shapes and associate it with things. And someone, yeah, Tracy, you can trace them out as well. But yeah, those kind of active learning of actually doing the activities or actually visualizing seeing it, we found those to be very effective in educating the kids on sickle cell anemia, and it just shows effectiveness of active learning for both within not just the primary school settings, but for the medical students as well. And again, when we were able to see them at the end, when they were showing like their parents and the other pupils in the presentations and how much they had actually taken in and all these little tasks that we've done, that's when we could actually evaluate that active learning that played a big role in just making it a bit of a successful program. So yeah, I don't know if I don't know, I cheated a little bit. I done a bit of a Blue Peter and I made some earlier. So I got my red blood cell and my sickle cell. So whenever anyone's ready, you can just put them up and you can have a look. That's just, again, it's just reinforcing all those active learning skills and things like that. So I'd like to thank the primary school children because they, again, were amazing. As Doa mentioned, we learned so much from the kids that you don't actually realize from how much they know and how much they can teach you. Because again, things like the normal blood cell, an easy way to describe to someone is like the shape of a donut, and we got that from one of the kids. So that we were also learning from them as well. And I'd like to thank all the medical students as well for all their help and also allowing me to help bring my skills forward. And thank you to Saeeda because without her, the whole program would have been possible, and I also would not have figured out exactly what I want to do in my life. I mean, that was also very helpful. So yeah, thank you all for listening. And any questions?
Fabulous. Wonderful. That was just so nice to finish with a kind of hands-on activity. Have we got any questions for our speakers? Oh, Paolo's still doodling. I'll maybe start with a question while we're waiting. You said at the beginning you wanted to kind of give people the opportunity to share with their camera off. Obviously, some people might be kind of reluctant to share what they've done just on pen and paper. Did you have any kind of reluctance from the kids, or were they just kind of willing to join in?
Do you want me to answer? I mean, I think Doha is actually a good one to answer that because she had the biggest group. So she had a group of, I think it was about maybe 5 or 6 children. Yeah, to be honest, when I was going around the different groups, I didn't see any child that wasn't willing to participate. There was one who didn't want to read, right? And I think that might just have been because he didn't like reading, but he was quite happy to colour in and draw and that kind of all those activities he was fine with. But the reading he didn't want to do that at all. But what was odd was that on the last day when we were presenting the posters, he was the first one to jump up. So I think he did okay, but I think Doha had the most there's a question in the chat about diversity in the classroom. Doha had the most diverse group. So I think, Doha, if you want to answer that one.
I think all of the children were just fine participating in different activities. The only child that was a bit reluctant was during the poster presentation. He didn't really want to say his parts in front of like the older children because he was embarrassed of like presenting, but he just needed a bit of motivation and just pushing. And like I helped him like present the poster presentation with him, where we weren't like supposed to actually present with them. They're part of the module, but I got to present with him, and that made him want to present. But other than that, all the children were fine with like just participating. I think being reluctant and embarrassed of others is just like something that us adults have, of just we're fearful of making mistakes or saying the wrong thing. But that's something I got to learn with the children, is just say what you want to say and do whatever you want to do, and you're gonna be fine. You don't need to be embarrassed of doing like different things if you want to do it.
Okay, fabulous.
I think someone said so I think there's a bit of part of that question as well, the diversity one, about like did anybody have any personal connection to sickle cell and just handling like sensitivity. I think we at the beginning, I think we did. There was a question asked like, did anyone know? Just when you were testing their knowledge, does anyone know anyone with sickle cell? And no one in the class did. But I think we did. There was a point we did go back after one of the sessions, and we kind of sat down and we were kind of talking about like the kind of languages we use and making sure we were very careful in how we explain things, and not because we never wanted anyone to ever come across someone with sickle cell and assume there's something wrong with them and use the wrong languages and go out. So I think that was one thing I remember we did after one session. We kind of got back together and everyone was talking. And again, I was just coming in from like an evaluating of how the process was going, making sure our languages were careful. You weren't making anyone feel like there was something wrong with them. It was just explaining this different concept to them and things like that. So just in terms of sensitivity.
And I think one of the other issues as well was that because when we did the Nigerian side of things, it's like Doha mentioned, this work is going to get taken to Nigeria and also delivered in Nigerian primary schools because they want to raise awareness. And Nigeria, when Testimony actually gave the presentation on Nigeria, she did say it the highest prevalence, but how we got by that as well was by saying it's in warmer climates. Right, so wherever there's a warmer climate, you're more likely to get something like sickle cell because that's where the highest levels of malaria are, and there's like a selective advantage there. So that was how we kind of got around that, and that concept made sense to the children as well, because I think, if I remember correctly, there was a child, he wasn't from, his heritage wasn't Nigerian, but I think he was from the area, but he actually said as well, it's in warmer places. And I think because it came from a child's mouth, it was easier to take it, to kind of go with that and develop that concept further, because it was a child that introduced that concept as well. So that's how we got by some of the sensitivity of it, because this was something that we were very aware of when we went in, actually, that we didn't want groups to be kind of labelled almost. So we were just very conscious of that.
Yeah, good. So such an impressive piece of work. It's just wonderful. Anyone else get any questions? I think we've got maybe a minute before we kind of, I think Paolo's going to close us for the festival, but any other questions for our presenters? I've got one. Has it made you want to go into teaching, Esther and Doha?
No.
No? Not in schools. What?
No, the kids were lovely. It was never anything to do with kids. I think I enjoyed the aspect of being able to come in and like teach a little bit and kind of go away. I think it didn't, we kind of reinforced that teaching others in some capacity, but not a full on, I'm going to become a teacher, sadly.
Okay.
Because that's the thing, you see how hard it is you have to keep the kids engaged, and it is a lot of work. And it's like, we got lucky and we're coming in for 5 weeks, if that, and we see them for a little bit. And you start to understand a bit more of how difficult it can be. So yeah, it was lovely to get that experience, but I don't think it made me want to turn become a teacher, sadly.
Okay, and what about higher education? Have you been inspired by Saeeda to think about maybe a future in higher education? Would that be maybe?
I don't know if Dola has to answer for that.
Honestly, before the SSC, I had no intention of doing any kind of teaching in the future, but now I'm more open to it. And I think more higher education than primary school, because like I'm not good at classroom management, honestly.
She was great, honestly. I think she's underestimating herself. She was really good. There's a question in the chat about assessing the degree of understanding among the children. That was actually through the presentations that the students did, and they had like inbuilt quizzes, and then we had the poster presentations as well. And alongside that, the children kept a glossary of terms. So each time it was in like hemoglobin. Many of them would just stand up and go, hemoglobin does this, it carries oxygen. And they were like starting to just say it and then talking about how the blood flowed in the red blood cells and the white blood cells. But I think one of the most impressive things actually that really kind of stuck out was one of the medical students actually had a little girl in her group, and they have one Friday a month where it's called Pajama Friday. So they all come dressed up in their pajamas. And this little girl brought in her bunny, right? And she introduced her bunny to her her medical student facilitator. And coincidentally, the medical student also had the same bunny. So they started having this conversation about bunnies, and then a little girl came up with the concept herself that her bunny could have sickle cell trait if the mummy bunny also had sickle cell trait, but the daddy bunny didn't, right? And she grasped that concept, and she explained the full thing, and she explains it so beautifully that one of the science communication activities that I have now is actually a family of bunnies, like the Sylvanian families, to kind of explain, right, we have these bunnies. So it's bringing it back to what Doha and Esther said. It's like you learn from the children as well. It's almost like a switch happens when you're older that kind of goes, but you need different perspectives and age groups to kind of remind you that there's different ways of learning. And that little girl explained it beautifully. And she recorded a little podcast on it, right, which isn't available yet, but it will be, right? So we're going to make this available, but she explains it in her podcast as well about the mummy bunny and the daddy bunny and having autosomal recessive inheritance. And it was just really impressive how much they actually grasp.
Yeah.
And if anyone's interested, we're going to be in the Gorbals Fair on June 7th, so you're welcome to join us. And they're presenting their work there.
Wonderful. And when you have that podcast, we'll add it to the festival Padlet as well. So let's just give our team, Saeeda, Esther and Doha, just a wonderful round of applause, because that was just excellent. And I think Paola's going to take us into the last part of the festival.